So everybody working on computational genomics have come from some certain expertise. And by training, I'm a statistician. In particular, I'm a Bayesian statistician. So I'm passionate. I feel like I have to tell you what the benefit of doing Bayesian, which is cool, in computational genomics. So, so this is the outline of the talk. So the talk is a little bit different than the regular tutorial. So I mix up some of my own research into the talk. So the benefit of doing that is so you can see that what you're gonna learn or you have already learned as basics is not that far detached from the research you're gonna do. So it's all based on very simple ideas and the right type of the thinking. So we'll first review the principle of Bayesian statistics, and I'll give a comment on model versus algorithm. The next topic will be the Bayesian hypothesis testing, which related to the multiple hypothesis testing. So if you have never come across that topic, that will be perfect because I'll give you the first crack on that topic. So for application, this is related to my own research. I'm talking about the EQTL discovery, so-called e-gene discovery, integrating genomic features. I will wrap it up with discussion and conclusions. So, well, let's start from the very obvious. What is Bayesian inference? So you may not do it in the research, but you certainly should do some homework or the Bayesian inference problem in your statistics class. So this Bayesian inference start with defining a parameter of interest and actually construct a parametric model. So when we talk about Bayesian models, this is my own view. We think the prior is part of the model, and then so is the likelihood. So you have a prior, you have a likelihood, you put everything together, and then you have a parametric model. So the example I'm going to use hopefully show that prior is actually not that special. It's part of the hierarchical model. And when you have the model, then the next task is actually doing the inference. So this is how the Bayesian got his name, the Bayes theorem. So you apply the Bayes theorem, which is a fundamental probability equation right here. And then you get this, the quantity, so-called posteriors. And that's posterior distribution of theta that contains the full information of the inference result. At least this is the Bayesian view. I didn't tell you how to use the posterior, the probability. So the next thing, maybe two slides, or trying to persuade you maybe. So why this is a kind of a appealing paradigm for inference. So a lot of people talk about Bayesian's get into the discussion of subjective probability versus objective. That's important, but really the Bayesian statistic appeal to me is because of the following two slides. The first slide is so-called likelihood principle. So this is basically when you do inference, you think about what is intuitive, what is right, what is not so obviously wrong, so to speak. So this likelihood principle essentially asserts the irrelevance of outcomes not actually observed. So if you are making inference based on the data, that's totally sensible. So if you're making inference based on something you could have observed but not actually observed, that sounds unintuitive, right? So not only Bayesian hold this view, the other kind of two well-accepted frequentist inference principles actually joint imply the likelihood principle. The first principle, the well-accepted principle is called the sufficiency principle. You should know that if you learn your statistics. So if you get your data and you have a parametric model, only the sufficient statistics matters. You can throw away the rest of your data. So this is the same sufficiency principle you learned. The second principle called the conditionality principle. So this is basically saying irrelevance of experiments not actually performed. So it's a little bit like we described, the data you could have observed but not actually observed. So that should be irrelevant, right? I mean, intuitively to your inference. This disclaimer is that right? This is showing up. The conditional likelihood, conditionality principle is used for inference. Has nothing to do with experimental design. So if you're doing experimental design, you should really consider the experiment not actually performed because you're doing that. So the other different perspective that could persuade you into the Bayesian camp is the definite theorem. So this is a go even early. There's the 1930s or 40s. There's a beautiful theorem about the probability. So the basic kind of a concept about this thing is, in statistics, if you learn a lot of like law of large numbers or the maximum likelihood estimation, so we'll start assuming the random variables are IID, meaning identically independently distributed. It's not necessarily intuitive. So let's consider kind of a thought experiment. If you flip a coin like 1,000 times, and then the usual thing you would do is every coin toss outcome is IID kind of observation, right? But if I tell you, if you observe 1,000 observations, they all turn out to be head. And I ask you the question, was the 1,001? And flip the same coin. If you really think that's independent, that's a little bit silly, right? I mean, there's just unintuitive. So that means is the first observation, the first set of the observation actually share some information, and then that's make the future observation becomes dependent. So what are they share information about is this definite theorem tells you. So instead of assume all the observations are actually IID, there is a more kind of a much weaker assumption, but it's a much powerful assumption, is saying they're exchangeable. So if you randomly swap the order of observations, you wouldn't notice. Every single observation just equally possible appear to any of the positions for finite sequence. And the definite theorem basically telling you, if you really have this sort of the exchangeable sequence, what you're going to have is you don't have the independence or something, but you have conditional independence, that conditional on that particular latent variable theta. So this, maybe you don't really need to understand the details of the theorem, but rather than saying that this has certain form of a prior and then conditional independence. So you make a much weaker assumption and then much more intuitive in general. And then this result, the definite theorem, gave rise to a thinking of latent variable modeling and hierarchical models. So hopefully this is the two things convince me that Bayesian is the right inference paradigm. Hopefully that they do the same thing to you. But the next, I'm going to walk you through a real genomic example, and then see how we can build a model from first principle. So the example I'm going to use is so-called a cis-EQTL mapping. So it's actually just identify so-called local genetic variants associated with expression phenotype. So this is it's not very different than genetic association study, except that you're focused on a molecular phenotype, which is the gene expression. And the cis part is just saying you're not interested in, well, it's at least for this task, the whole genome genetic variants. Rather, the genetic variants that is local or close by that particular gene, the target gene. So, well, as I said, this is not very different than just the regular genetic association study. So we can model the genotype-phenotype association using a multiple linear regression model for each gene L, which is denoted. So the formula is really not that complicated. Rather, I want you pay attention here is just we are actually trying to do the job without thinking if we can fit this model. We consider OP different markers could potentially associate with the expression, which is denoted by y of L. And so it's a general model. Even the model, even the SNP is not associated. The beta L is simply here is 0, right? I mean, that's the model is still right. So it's kind of a generalization to write down the association model. So I'm going to argue for the interest of cis-EQTL mapping. We're not really interested in the effect size. Well, I have to say, there's some of the studies interested in the effect size, but not in here. We're interested in identify the dichotomized beta. So if the beta is 0 or non-zero. Okay, so this is basically what you do for hypothesis testing. You're going to make a decision if this particular marker is associated or not associated, if that's easy to understand. So the reason is the beta is not really interpretable in this context. Is that in the genomic, especially RNA-seq, we do quite a bit of transformation, and then latent factor control that the Y itself is not really represent the expression level, at least easily. Okay, so this is a part is the likelihood, so everybody should agree, I mean, as approximation at least. So the thing, the interesting part of this model is we're trying to incorporate some of the genomic features like annotations of the SNP into this type of the model. The reason is very simple. So if you know a priori a particular marker going to disrupt the transcription factor binding, then it's very much likely that this marker going to affect the downstream, the target gene expression. That's sort of the simple, naive thought based on the biology we learn. A very simple way to put that thought into a mathematical formulation is actually write down this logistic model and then saying, okay, so if the gamma is we have defined in the previous slides, the odds of gamma are going to be higher if the particular SNP or the variant have certain features, right? So that's denoted by the D. So that's a feature. For example, binary feature is this SNP is transcription factor binding site or not, 0, 1. And then in this case, we introduce another set of the parameters, alpha. So alpha 0, alpha 1 refers as an enrichment parameter. Especially if alpha 1, if it's positive, that means having this feature will increase my odds, increase this particular SNP's odds being the causal SNP. If it's 0, that means this feature has nothing to do with particular SNP being a EQTL or not. If it's negative, that's obviously saying the depleted, right? That type of the situation. And the key thing is this parameter is sort of shared among all the gene SNPs. So this is sort of think about the Di-Finetti theorem. That's the latent factor shared by all observations. That's we can really learn from the data. So that's the beauty of this thing. To complete a Bayesian model, we have to further assume a priori independence of gamma. This is a model choice, and it would be a mistake to say you have to assume that. But if you don't assume that, you're just. But there is no particular reason a priori you're going to say that the gammas are non-independent, right? And we will also assign standard priors for the other parameters, the beta, mu, and then sigma square. So I say standard because so this type of the priors people have studied very extensively. So they know that when you have large enough sample size, the priors on these parameters are not going to change the inference result quite a lot, let's say. It's quite stable under this type of the prior. So one important thing I want you to notice is that the full model here we just built up is generative, meaning that we can simulate data from it, right? I mean, if you want to simulate it, you just take this model and then draw random variables like gamma first, and then draw beta and the mu, and then you can simulate from the linear, multiple linear regression, and then actually compare your simulated data with the observed data, see if this model is a good one. So that's kind of the idea. We're going to come back to this point a little bit later on. So to finish the first the part of the talk, the introduction to Bayesian, is the Bayesian computation. So this is really the bottleneck of applying Bayesian in the genomics. The exact inference typically not existing, except a few very naive examples. The reason is if you look at the Bayes theorem, the denominator, go back to this slide. Oh, sorry, there's the wrong direction. So the right-hand side, the denominator, you need to calculate an integral. So calculating integral is much harder than typically much harder than optimization problem. So that makes the Bayesian computation is really difficult. So there are common solutions rely on generic numerical recipe based on the sampling-based approaches. So if you have heard the Markov chain Monte Carlo type of the approach. So those things, the most important thing you need to understand, those are approximate algorithms. So they give you approximation of the results. Because the whole thing typically, the exact calculation is an NP problem. Since we haven't resolved the NP complete problem, so there is no exact solution that make the whole computation scalable in a reasonable time. So the computational challenge is really for us to practice Bayesian statistics in this area. We should care about the efficiency of the computing, and then we should care about the scalability of your proposed method. There are a few comments on model versus algorithm. So from Bayesian point of view, so we should give a rather kind of my own definition of a probabilistic generative model. So this is based on what we have done for the CCQTL model. So this is just a set of assumptions that hopefully can be used to simulate realistic data. So there has nothing to do fit the model. Okay, so all the things I said are assumptions, and then you can use that to simulate. So the benefit of doing that is you can actually check your model, because as I said before, you can simulate the model from your probabilistic assumptions and then compare with the real model. So this is a so-called Bayesian model checking. It's quite powerful approach if you have doubts, if you think you're making some crazy assumptions or things like that. The algorithm, on the other hand, is a computational procedure for model fitting. Okay, so you have a model, you have an assumption, so the algorithm will just tell you how to fit the model. So that's my view. So good practice is if you present a computational approach, you should try at least present model and algorithm separately, because that puts some of the assumption up front. You can check, and then you can know in principle sometimes when some assumption broken, your computational approach cannot be applied. But maybe it's difficult. Sorry, it could be difficult in practice. Okay, so as an example, I'm showing you a kind of again, there's an empirical base as an alternative algorithm view. So back to the CCQTL mapping example. So we didn't actually mention any of the priors put onto the enrichment parameter alphas, right? So if you think like Bayesian paradigm, you could do this endlessly. So you build hierarchy after hierarchy, because in principle, every parameter you need to put a prior. So there is endless kind of a loop here. But what we could do here is we simply attempt to compute and plug in this MLE alpha hat for the computation. The reason for that is the data you have going to be pooling across all the genes. So you have quite high information about these two parameters just intuitively. So you can do a good job in terms of estimating alpha hat. But the alternative way to see this is a really kind of approximation for true Bayesian inference, which requires you to integrate out alpha, so to speak. So I see that as an algorithm kind of approach to shortcut or cut the corners for the computation, rather than this is rather assumption. So just give you that example for that. All right, so I'm going to switch gear to multiple hypothesis testing and then go back to the cis-EQTL example. So if you never, as I said, if you never heard of multiple hypothesis testing, that would be perfect. If you learned about the p-value and the Benjamini-Hochberger, that's great, but I want you to forget that for a minute. As anything in Bayesian statistics, we have a model. It's rather a general model on multiple hypothesis testing. So the model is actually quite simple. So we assume there are going to be m different tests. For each test, we're going to get a set of data denoted by y of i. I runs from 1 to m. And some of the yi's come from null model. So this is denoted by the z of i, right? Z of i is binary. If it's a 0, if z of i is 0, that means the data from the ith hypothesis testing is generated from the null model. Otherwise, the zi equal to 1, that means it's actually generated from the alternative model. So there is no restriction on p of I. So each different tests can have completely different null model and an alternative model. So it seems like they're completely independent. But there is some commonality. Just connect all the tests together. The view of this is z of I. You can view them as they are kind of a coin toss from an independent Bernoulli distribution, denoted 1 minus pi 0. So this is basically the idea. Again, the definitive theorem. This is a latent variable that connect everything together, right? So they are sharing information about how many, what is the proportion of the null hypothesis in the overall type of the data I'm going to observe. That has a very important implication for the decision you're going to make. If the pi 0 is close to 1, then obviously you need to be overall very conservative. Something like Banferroni correction should be applied. But if the pi 0 close to 0, so almost all the tests could be true, right? You should be allowed to be a little bit liberal in terms of rejecting the null hypothesis. So that's the idea. Are we all clear about this model? Any questions? That's kind of important. All right. So the y's are typically, I should say, this is not the Bayesian view of the whole thing. This is actually a general, this is a view is shared by many frequentist approach as well. So the y's are assumed conditional independence, the conditional on pi 0. So what we do in terms of hypothesis testing is actually making decisions for each z of I, right? So we observe all the data, and then we put all the data into kind of a decision function that's denoted by delta. And then delta spit out either 0 or 1. Think about that. You're actually doing it if you're using p-value. So you compute the p-value using your y. And then you have a threshold, and then you say, OK, if this is lower, if the p-value is more stringent than the predefined threshold, I'm going to reject it, saying this is 1. The delta is 1. Otherwise, it's 0. So you're not this is not very different. It's just a formulation. And then we can define the total rejections by sum over all the decisions together. Remember, there's a binary, so the sum of these is from OM tests is essentially the total number of rejections. All right, so when we have a lot of tests to do, the obvious question is, what should we control? So one way to control is so-called family-wise error rate. So that's saying, all right, we do a lot of tests, and I don't want to afford to make a single mistake. That thought is a little bit stringent, and then sometimes you feel like unintuitive. What's the reason you do thousands of tests and and then you cannot afford making one single test? If that's really not it's your goal, what you should do is just go home, do nothing, and you're guaranteed not making any mistakes. So the kind of a glass half full type of the view is you have more tests performed. You're going to find more true things, right? And then that's actually an opportunity, and then to find more things. So the reasonable thing to control for, I think, is arguably is the proportion of the false discovery. So how do we define that? So this is an important slide. It's define the false discovery proportion, which is so this is a little bit complicated, but if you really read into the math, the numerator is basically the false discoveries. If the delta is 1, and then the zi is 1, so you make a right decision, that doesn't contribute anything to the numerator. Only if the delta is 1 and the zi is 0, so you make really the type 1 error, that the particular test contribute 1 into the numerator. And then the d here is, as we said, is the total kind of a rejection. So this is a very intuitive definition of false discovery proportion. Okay, so next thing is Bayesian FDR. As we said, based on the likelihood principle, we want to control this FDP, which is a random variable. You have to realize that. Is we conditional on all observed data and then compute expectation of that FDP. But if you do that, you find this computation is actually easy because delta here is a function of observed data. So if you condition on the data, it's no longer a random variable. And then the only thing that is random is z in that the FDP definition. So from the first line to the second line, the only thing differs is I replace the z of I into a probability, which is probability z of I conditional on data, which have the form of posterior probability, right? So that's Bayesian FDR. It's so simple, right? I mean, it's very intuitive. And then there is also correlated to the frequentist FDR. As we said, the frequentist FDR is going to be take another expectation with respect to all the data y. So it's kind of a thinking, if I hypothetically assume I collect the more observed the data, what's going to happen? So it's the double expectation on the last line. But if you do that, that formula is not easy to compute because y becomes a random variable. And then if you attempt to directly compute expectation of FDP by integrating out both z and y, it's non-trivial. Let's say that. All right, so for Bayesian FDR, it's so easy. So we just need to compute. So this is basically the last slide. So it's a natural decision rule to use the posterior probability, actually compute the, this thing keeps popping up, to make the decision. So this rule is a lot like the p-value rule. Instead of using p-value, here we can compute the posterior probability and then compute the expected posterior false discovery probability, which is 1 minus v of I. So this, remember v of I from the previous slides. Everything you need to do is really compute the posterior probability of z of I. And then there is optimal procedure. I don't want to say too much. Again, this looks intimidating, but it's rather than just saying you can sort all the false discovery probability together from small to the largest, and then find the pool, find the section of the sets of the hypothesis which you have average, the partial average of those probability is less than or equal to alpha, so you control everything. So it's very simple. The take-home message is you just need to compute the probability of zi given y, and then you're done for the Bayesian FDR control. All right, so hopefully you feel like that's natural. I mean, that's the key word here throughout this talk. So we're going to consider a problem, get back to the CC-QTL problem. We're going to ask the question is identify genes harboring at least one eQTL in its cis region, okay? So there's many kind of statistical questions we ask using the eQTL, CC-QTL data. So how do we formulate this into a statistical problem? That's the interesting thing. So we can easily formulate that as a hypothesis testing problem. So for L equal to 1 to capital L, that's the number of genes we have, we just need to test the null hypothesis stating none of the cis SNPs for the gene L associated with the target gene expression, which is just the gamma L equal to 0, right? That's a very straightforward null hypothesis. However, we need to do that for tens of thousands of genes simultaneously. That's why there's kind of a multiple hypothesis testing problem and very naturally arise here. All right. OK, so before we go to the Bayesian solution, we can discuss a little bit of the non-Bayesian solution. So this is kind of an algorithm description. So this is typically you do in read the journal. You don't really get not everybody present the models in a way as Bayesian, but I mean they present how they do the procedure. In the view, it's an algorithm. So, but this algorithm is very intuitive. So what they do is they find the strongest single SNP association signal for each gene. For example, the maximum z-square, the smallest p-value, there is essentially the same thing among all cis candidate SNPs. So that statistic is regarded as a gene level test statistic. Then to deal with multiple hypothesis testing, in a frequentist way, you have to convert that into a p-value. But it's not easy, I will just say that, because of extensive linkage disequilibrium. So what you do is, typical solution is permutation. And there are also other much smarter approaches can avoid the permutation, like Eliezer's group developed. But I mean, still, it's computationally intensive, as you can imagine. And then we can gather all the gene level p-value. Now they are really legit p-value, and then put together to perform frequentist FDR control. So in this sense, if you know Benjamini, Hochberg, or Story's the Q-value approach, that will be along the line. So there's the procedure you're going to perform. So there are some drawbacks, obviously. The first thing is I mentioned there's computational efficiency. The second, I think, the drawback is it's not fully compatible with our parametric model, because there's no obvious way to incorporate the, at least for now. I mean, there are ways, but I mean, there's no obvious extension to this procedure incorporate the genomic annotations, right? So for Bayesian solution, it's conceptually really easy. I'm just trying to convince you that. So the previously constructed Bayesian hierarchical model is a generalization for both null and alternative hypothesis. As I said, so if you set all the beta at 0, that's basically null. And then if there's something is non-zero, that's alternative. So it's both covered, both scenarios are covered in a parametric way. And we have shown the model also kind of naturally incorporate genomic annotations. So from the kind of the introduction to Bayesian FDR control, what we just need to compute is this particular posterior quantity for each gene, the probability of gamma L equal to 0 given all the expression genotype data, right? That's basically putting everything together, compute the posterior gamma L equal to 0. So as I said, this seems like a straightforward recipe, but you're going to run into computational troubles. This one is not really computable if you just try brute force. So we're going to apply some approximation. So this is sort of, my view is kind of algorithm approximation. So first approximation, we're going to use empirical Bayes. Instead of compute the second line, the gamma L, given all the genotype expression data, we're going to compute for each gene that much simpler probability, gamma L, given the genotype, phenotype, and then the estimated enrichment parameter. So think about the alpha hat is actually estimated by pulling all the data together. So it's sort of all the other genes jointly gonna influence on the alpha hat, right? I mean, that's how you do the estimation. That's basically the power of this type of approach. And because I'm short of time, I'm just wavingly say that the MLE of alpha hat can be obtained through the EM algorithm. So the view on this is you just treat all the gammas as missing data and then write out the complete data likelihood and then do a straightforward EM. Then if you agree with this, then you are gonna ending up compute the last line. But I'm gonna say the last line is still not computable. The denominator, so the numerator is easy because we have for any, so this is just compute the gamma equal to L, and then the marginal likelihood of that particular null model. That's relatively easy. The denominator, individual terms, trust my word, is easy to compute. The problem is you need to sum over all possible gammas. So if you have p different SNPs, the all possible gamma values is 2 to the p. So very typical, we have thousands or 4,000 of markers. So think about sum over 2 to the p. So it's impossible, at least in a reasonable sense. So this is our kind of approach to tackle this problem. So we basically find that sparse models dominate the posterior probability mass. What that means is if you look at all the identified EQTL, you don't find a lot of associated SNPs. Typically, you find there probably one SNP associated in the whole cis region, right? That's very different than need to enumerate all the gammas. So maybe you can just basically scan a relatively low dimensional model in terms of like very sparse model. That will reduce the computation dramatically. So that's basically the idea. That's motivated by this observation. So majority of E genes, only a single SNP signal identified. So if you can do that, we have an algorithm, it's called a DEP1. It's in brief for deterministic approximation of posteriors. The one means you just sum over all the single association model. So that's basically what we write. So instead of evaluate all possible, the so-called normalizing constant, which is, sorry about that, the denominator, we're going to calculate a much smaller space and then assume kind of this space can occupy majority of the probability, posterior probability mass, which in practice is quite good. So with this approximation, you can even do approximate posterior calculation analytically. So there's just kind of a day and night difference between like you need to sum over 2 to the p models, or you compute. Well, I'm not saying that you can do it in the pencil and then paper, but it's pretty close to that. And then the other benefit is utilizing only single SNP association statistics. So to put this in practice, what kind of a feature we can use? So if you think the feature that can boost the e-gene discovery most must be the feature that enriched the most in the eQTL. So that's just again, this is a natural intuition. So this is everybody find by doing cis-eQTL discovery. The number of eQTLs is highly correlated, their distance to the transcription start site. This is a much more smooth version we did in our paper, but there's different ways. If you plot the p-value, you get the same pattern. And then it's dramatically kind of flat out when the distance goes to beyond 100 kb away. So this is kind of information everybody find, and then there's a kind of a very strong enrichment pattern, and then we can use it. If we use it, first we have to make sure we can estimate this type of the decay very well. And this is some simulation trying to show. So we simulate the right line is kind of an exponential decay, but it's a modest exponential decay. And then the histogram is estimated by the EM algorithm. It's not perfect because we have to bin the SNPs into each bin. So there, you see that this unequal size bin showing in the bar, that's basically you bin the SNPs into the blocks. And then for each block, you estimate the probability, the prior probability that, that in that particular bin, the SNP associated with the gene expression phenotype. So it's not perfect, especially you zoom into the peak, but overall it's captured that pattern. So the other kind of a simulation trying to show it's a much rapid decay, and we can still capture. So this one is actually more close to the real data, and just let you know. So we try this into it's basically kind of the showcase of this type of the method. If you utilize this information and then trying to identify e-genes and then compare with the standard method, what do you find? So this is this called the GTAX data, which is the NIH-funded project, just trying to mapping eQTLs in different human tissues. So the number of the points here represent different tissue type. Because they don't all have different sample size, so you don't really find the same number of the e-genes, or not even approximately similar number of e-genes. So the sample size in this particular data set varies from like 70 to 350. Typically, you have more sample, you find more e-genes. So this is trying to contrast that the y-axis is if you're doing the Bayesian approach without any annotation, versus you do the frequentist approach, like this is actually based on the Story's Q-value approach. So you see there's performance is almost similar. The Bayesian approach is even a little bit conservative for the reason we know, because the Bayesian FDR in principle is a little bit conservative, because you can take the frequentist one is taking another expectation, if you know what I mean. If you don't, that's all right. But the really benefit of doing this is if you really incorporate the number of the annotation, that is SNP distance to the transcription starting site, you see a uniformly increase of e-gene discovery. It's quite dramatic because, as we said, this is a very strong enrichment signal, and that's expected. So the Bayesian approach finds more e-genes uniformly across all 44 tissues. And then there's the Bayesian approach, again, I have to say, it's much more efficient. for the reason we applied the different kind of approximations. So on average, just give you a number, there's almost 1,400, close to 1,500 e-genes discovered. Remember, for most of the tissues, the total discovery is around 5,000 or so. And then there's some consistency, obviously. So there is a software implement if you're interested in this work. OK, so I think I'm actually a little bit over my time, but I'm just give a quick wrap up of the whole tutorial. So I hope you think more of Bayesian parametric modeling, especially explicitly state your assumption up front, even if you're doing a frequentist approach. The other thing I don't have time to discuss is as long as parametric model are utilized, the inference result on Bayesian and frequentist approaches are typically similar. So this is shown by the Wakefield 2009 paper, compare base factor versus p-value. So that's that paper is showing on the link on the Institute web. And the last thing is the Bayesian hierarchical model is very natural to integrate multiple sources of data. So this becomes extremely attractive in statistical genomics. So finally, this is some relevant literatures if you're interested in to get more details of this. All right, that's all. Thank you very much for your attention.